Gene Therapy for HIV by Josh Grams and Tia Oberoi. Gene therapy is defined as the genetic modification of somatic cells to correct a disease phenotype or to achieve a therapeutic benefit. As you can see in our little diagram, this is an example of gene addition, where added DNA allows the cell to make a therapeutic protein which can either cause a disease phenotype not to be shown or inhibit a disease from being contracted. The FDA defines three main mechanisms for gene therapy. Replacement, where we replace disease-causing genes with a better or healthier gene. Inactivation, where the gene is inactivated. And addition, the addition of new or modified genes to help treat disease. This is done through various different processes, some of which we will cover further today. The first being the addition of plasmid DNA, which facilitates the addition of a transgene into the cell. Another way is through viral vectors. Viruses are able to transmit genetic material into cells as their main function. So by modifying the genetic material itself, we can use them as a vector to add positive genes into the cells. A similar thing can be done through the use of bacterial vectors. Gene editing, which can be done both in and outside of the patient's body, is used to repair damaged or mutated genes.
Human immunodeficiency virus, also known as HIV, is a sexually transmitted infection that can decrease the patient's ability to fight infections and other diseases. HIV is transferred through bodily fluids. Without proper treatment, HIV can weaken the patient's immune system to the point of acquired immunodeficiency syndrome, also known as AIDS. The patient's immune system is weakened by the infection destroying CD4 T cells, which are white blood cells. Though there is no cure for HIV as of right now, there are many ways to control the infection. One of these ways is through gene therapy. One approach to gene therapy is the RNA approach, which uses ribozymes. Ribozymes are RNA molecules that have catalytic activity. These molecules bind to the antisense sequence of an RNA and cleave the phosphodiester backbone of that sequence. The cleavage of an RNA sequence can cause a change in the function of the RNA. Ribozymes are not always the best option due to their lack of adaptability. For example, if there is a mutation on the target region of a ribozyme, it may not be able to bind that sequence. If it is unable to bind, then it is unable to cleave that sequence, causing no change in the RNA. One way developed to avoid this shortcoming is the use of multi-ribozymes, which are collections of ribozymes that have similar but different target regions. RNA approach implications for HIV. Initial studies of HIV ribozymes show that they are able to cleave both incoming HIV genomic RNA and newly transcribed viral mRNA. This slows down HIV viral protein synthesis, decreasing viral expression and spread through the body. Other newer studies have developed more precise and adaptable ribozymes that are able to prevent HIV expression overall for up to 35 days after the therapy. The benefit of ribozyme therapy is the ability to identify and cleave at the leader sequence. What is a leader sequence, you may ask? A leader sequence is just a sequence conserved through all HIV strains, making ribozymes effective against multiple strains. This ensures that no more than one therapy is required for consistent prevention or suppression.
Another gene therapy that has shown much promise is adeno-associated viruses. So what is an adeno-associated virus? It's a non-enveloped virus containing single-stranded DNA, and they require another virus in order to infect their host. So what does it mean when it says that it's non-enveloped? It means that there are no membranes surrounding the genes themselves, which makes them very easy to adapt and to change. So, inverted terminal repeat sequences surround the two coding regions known as the rep and cap gene sections. These are the replaced gene sequences by the transgene, which is going to be what we want to be expressed within the cell. Adeno-associated viruses can integrate into the cellular genome and then be expressed through that. So why would we use adeno-associated viruses? Well, they are not associated with any known diseases and are very rarely known to cause any sort of infection due to their need to be an associated virus. That means that the host is rarely affected by this virus and there are no known side effects. So how does this tie into HIV? A recent study at OHSU has shown this new monoclonal antibody, leronlimab, to show great success in preventing the spread and adaptation of HIV within the body. So what is leronlimab? It's a monoclonal antibody, which are unique white blood cells that are specific to a disease that they prevent, that has been shown to stop HIV from entering immune cells. So this gene sequence is inserted into the adeno-associated virus and is injected into the host and is expected to proliferate in muscle cells to continuously make this antibody. In this project, researchers have designed a synthetic AAV vector, which is able to enable long-term production of leronlimab inside the body. This is an amazing feat because it shows a single injection that can suppress HIV replication and eliminates the need for long-term antiretroviral therapy.
In conclusion, gene therapy has shown to be a promising way to treat diseases such as HIV. Gene therapy, due to its adaptability and emergence as a new field, has many different approaches, some of which we covered. Of the approaches we have outlined today, have only emerged in the last 10 years, showing the ability for growth with the development of new technology, which is happening every day. We hope you enjoyed our presentation and are hopeful for the treatments possible. Thank you for your time.